From the Cosgun Lab and the Bioengineering Media Lab at Georgia Tech, welcome to Lab Coat Conversations, where you too can join the discussion on the next academic breakthroughs. Join your host, Hector Torres, as we talk to Georgia Tech researchers. This episode, please welcome Erin Buckley. So we are talking today with Dr. Erin Buckley, right, part of the Buckley Lab. Their mission statement is to develop and explore the clinical value of novel diffuse optical techniques for non-invasive, low-cost space monitoring of hemodynamics in brain and other tissues. So that's their mission statement directly from their lab. So if you could just tell us a little bit more about your lab and maybe what your lab focuses on a bit more to give the audience a little bit of context, that would be great.
Sure. So my lab develops tools that use light to study the brain non-invasively in humans. So the technology that we use is very similar to the pulse ox that they put on your finger at the doctor's office to measure your heart rate and your oxygen saturation. Only we are basically making a pulse oximeter for the brain. So we're developing this pulse oximeter for the head, where the idea is that your human tissue has this unique window of relatively low absorption and in the near infrared region, wherein light is able to penetrate very deeply before being absorbed. And the tissue is also very highly scattering in the near infrared region. And so it allows for us to deliver light at the tissue surface and actually be able to collect some light as the light gets scattered back to the tissue surface. And based on the properties of the light that we detect, we can infer information about how much oxygen's in your blood, how well your tissue is utilizing that oxygen, and how fast your blood is moving. And all of those things can be very clinically useful biomarkers of disease progression or injury, detection or response to therapeutic interventions after injury. So basically we use these optical tools. We develop that. We're always innovating on them in the lab, and then we're very interested in applying them to different populations that could benefit from a non-invasive tool that like ours. It has lots of different attributes, including being low cost and bedside capable, and so you can intuitively kind of think of many different clinical applications where a tool like that would be valuable.
Yeah, that's great. So I do research on everybody beforehand, right? But I'm looking at this one question, maybe you can help me out with it. How has your lab progressed the field, especially in terms of NIRS and DCS? I don't know where I necessarily found those terms. So if you could refresh me with a reminder of what they are too.
So the tools that we use, these near infrared light-based tools are kind of collectively called diffuse optical spectroscopies. At least that's what I call them. That's like the field of study. And then we specifically use two types of diffuse optical spectroscopies. One's called NIRS, NIRS, and one's called DCS, which stands for diffuse correlation spectroscopy. So, and NIRS is near infrared spectroscopy. So I see my lab as really pushing the validation and clinical implementation of these tools and demonstrating their clinical value. So I've actually been working in the NIRS and DCS space since I was a young graduate student. So usually they tell people in academia to do their PhD in X and their postdoctoral training in Y, and then you're faculty position, you can combine X and Y in some way that nobody's ever done before. And for me, I did my PhD in X being NIRS and DCS. And then I did my postdoc in NIRS and DCS. And then I did my faculty position in NIRS and DCS. So I've been doing it for a long time. And kind of generally my main push is on these clinical brain applications of the technology.
Yeah, that's great. So I guess in terms of that, how has your lab been able to like progress those fields in the real world?
Yeah, I mean, we do lots of different things, right?
Yeah.
So I could pick one, or
Yeah, just give us like one example.
sure. So one of my main interests when I came here, when I started my lab, was in applying the tool to sickle cell disease. So for those who are listening and don't know, sickle cell disease is a blood disorder that's associated with basically loss of red blood cells, anemia, which has profound consequences on every organ in your body because all your organs need blood to survive, right? So the children and adults with sickle cell disease have a very high risk of stroke. And I saw value in the technology. So first of all, I guess for context, Children's Healthcare of Atlanta sees the largest cohort of kids with sickle cell disease in the country. They see over 2,000 kids a year with the disease. And I saw an opportunity to merge our technology with the amazing clinical environment that we had here. So I think that, especially, I'm going to talk a lot about kids because we work with Children's Healthcare of Atlanta, but these kids could really benefit from a kind of point of care device that could alert them of times of impending, I guess the fancy word would be cerebral hemodynamic crisis, but really like times when their brain is starved for oxygen and they need to get to the clinic and either get a blood transfusion or kind of be assessed for risk of stroke. And they don't really have anything at the moment like that. And so we've been working for the past many years to just demonstrate that the tool is valuable in sickle cell disease, one, that it works, and then which we've pretty robustly done at this point. I think we've made great strides in that area. And then two, that it can actually be a useful clinical biomarker. And that's what we're working on right now. And then along the way, so I'm a physicist by training, and one of my interests is in how light tissue interactions and better understanding that. And one of the techniques that we use, DCS, we know that it works. We know that it measures blood flow in the tissue, but there's still a lot of questions as to how it works, I guess I would say. And so I think sickle cell disease is a really interesting platform to study how it works, because sickle cell disease alters not only the concentration of red blood cells, but also their size and their rheological properties. And so we've made a lot of strides in developing kind of tissue simulating phantoms. So tissue simulating devices that we can in a very controlled way, study how altered things like blood cell concentration influence our optical signal, and developing models to correct for those effects to better improve our accuracy of our measurements in this population. So, we've come a long way and we've made a lot of progress and published many. I was just writing a progress report on this grant. We've published many papers in this space, but there's still a lot of progress to be made in this space as well.
So in terms of that newer progress, do you see any like newer imaging technologies coming into your lab, or maybe newer
Yeah,
concepts that you're trying to explore?
absolutely. So, I mean, the main challenge with this approach is that it's non-invasive. So, the light has to, when you're applying it to the brain, you have to make it through the skull and the scalp before you can get to the brain. And then the light has to go back through those layers to get back to the tissue surface. And so the light that we detect has a lot of what we call extra cerebral or skull and scalp contamination to the signal. And the name of the game in our field is minimizing or removing those contributions to the signal. We have done a lot. My lab has done a lot in the kind of post-processing side of things to enhance our modeling approaches to remove those contributions. But in the past 3 years or so, there's been a lot of hardware approaches that have been coming out as well, that it basically enhance depth sensitivity of the technique so we can probe deeper. And we have started implementing some of them in our lab, and I'm excited. They all have their, pros and cons. And I think the spin that we'll bring to it is finding the approach that can be best suited to the clinic and actually implemented clinically.
That's great. That's really interesting to know about. In terms of your lab, do you have any advice to give to students who are trying to find a lab or who are interested in your lab specifically?
So in general, my advice to young students is, I don't know. For me, I can get excited about any problem. I think as scientists, we generally all can get excited about most unanswered questions. And so I think the important thing is, one, to find a problem that you are excited by, and then two, to find a lab environment that you can thrive in. So that means both a PI whose mentoring style aligns with your needs from a mentor, as well as the remainder of the trainees in the lab. You need to click with them because you're about to spend, for PhD students, the next 5 to 7 years of your life with those people. And they're lasting connections that you'll want to take, throughout your academic or professional career. So I think the choice of lab is super important. We do a lot to make sure that the people we choose mesh with the environment of our lab.
So you mentioned PI mentorship style. Do you specifically have one in mind that works for you?
Like if trainees want to work in my lab,
Yeah.
what kind of trainee do I look for?
Yeah.
So for my lab and for me, I think people who are so I first and foremost, I look for people who are computationally savvy because we do a lot of data analysis, and we synthesize large data sets and stuff. But beyond that, I'm looking for people who can who are team players. Everything we do in my lab is collaborative because it takes a small village to make these clinical studies happen. I also look for people who have personalities that will kind of seamlessly integrate into the clinical environment, because that is a very stressful environment. And, yeah. And then I look for people who can, I guess, juggle many projects at once. So people in my lab typically don't just have one thing they're working on. They may have one large thing, but then they have like 3 or 4 sub projects that they're progressing on. So I look for people who can, who like to do that, who like to switch from thing to thing and keep busy.
That's great. Thank you so much for all the information, and thank you so much for taking the time out of your day to meet with
Oh,
me.
of course.
I have one last question for you, which is, where can students find you if they have any more questions?
The best thing would be to email me. We also have on our website, we have like a link that says, so buckleylab.georgetech.edu, or it's actually buckleylab.gatech.edu. And then we have a join the team tab on our website. So that's mostly for undergrad or master's students, but PhDs could fill it out. But really, you could just email me, and I may be slow to respond, but you just keep emailing me and I will write you back. I never mind being nagged, is what I always tell students.
All right, perfect. Thank you so much for all that information.
Yeah, of course.
Great.